So I'm very glad to be here, because in my daily life I don't have that many opportunities to be close to younger students, and it's always very inspiring. So thank you very much for the invitation. And that made me think when I was doing my PhD, and that was like 20 years ago, and it was the beginning of lab on a chip, what we call a microfluidics, what is now the point of care systems. And like nobody really understood what I was doing. I remember like my mother, like at the end, she arrived to the conclusion I was doing a pregnancy test. And like, good, like we will talk about pregnancy tests because they have a key role in all the story, but like she really killed the innovation of my thesis with her conclusion. But nowadays, diagnostics is in a very different place. And this is what I wanna share with you, and also hear about your thoughts. So like what is out there and how we arrived here. So in order to define what is in vitro diagnostics, we need to do one step back and first explain what is medical devices. So the medical devices are instruments, tools, softwares that they can work alone or in combination with others to assess a medical condition, right, with a medical purpose. And I put some examples in here, and you can see, like, for example, one of the purpose can be supporting life, and that will be like a heart pacemaker. Another could be diagnosis, and put an image of a scan that could be used to find any tumor or any wrong thing in one of our organs. Then we have monitoring. A lot of the population has high blood pressure, especially elderly people, and they need a device that can help them to monitor their blood pressure and control if the medication they are taking it is doing the job. And finally, also, they can be used for treatment, the scanner here, using cancer on an MRI. So then in vitro diagnostics is a subgroup of the medical diagnostics. And basically what they do is through samples from our body, and it can be all type of samples, blood, sputum, vaginal discharge, urine, any fluid that comes from our body, and use it to diagnose a health condition, right? And the difference between in vitro diagnostics and medical devices is that the in vitro diagnostics are never in contact directly with the human body. So in vitro means on a glass. So basically we take the sample outside of the body and then we do the assessment. And I also put like 4 different examples of the purpose of diagnostics, of in vitro diagnostics, sorry. And one, it would be the diagnosis, and that's easy. And it will be, for example, if we test if we are having flu. We want to know, yes or no, do we have this infection? The other one could be disease progression. Imagine you have liver condition. You can measure different enzymes and analyze. Is it anything? Sorry. Yeah, sure. Can we just move the table? Okay, that's it. Now we lost. All right, you're back. Yeah, thank you. Sorry, it seems I was out of camera. I don't like to be the center of attention. Okay, now I lost my track. All right, yeah, we were talking about disease progression. For liver disease, for example, yeah, there are a lot of enzymes like ALT, ALP, the bilirubin protein that we can monitor how the levels are, and then have like an understanding of our liver condition. So that will be an example of disease progression. Predisposition of a health condition like cancer, diseases that they can be transferred from parents to children. And the last one, it will be it will involve a therapeutic decision. Imagine like for diabetes, like the measure of the glucose levels in the blood will determine the amount of insulin that you have to take. And when do you think that the first diagnostic was used or discovered? And actually, I think that when I was preparing this talk is one of the first times I really reflect on that, and I have to look for it. And I created this slide where I try to capture the main 10 success events that I think shaped diagnostics until now. And what I found is that it was already used in the ancient times, where physicians observe, for example, urine to determine the condition of a patient. And in this case, it was not through techniques. It was more like observation and relate the observations with the symptoms, right? But a lot of the times when they could see something in the urine, the disease was already quite advanced. So it really lacks of sensitivity and a specificity. And then it was like in the 60s, 70s when the microscope was discovered. That was like a big change. We moved from observations of symptoms to actually be able to see cells and pathogens. And we moved from the macro level to the micro level. Like a century after, we enter what I call chemical era. In this period of time, it was already like the quantification of proteins was possible. And if we keep the example of the kidney disease, then proteins were able to be quantified and related to the symptoms. So it was when it started a bit the idea of laboratory testing with quantification. Then in 1859, radio immunoassays, they were discovered. And this is like antigens and antibodies. They were discovered to be able to detect molecules. And it was the beginning of the immunoassays. And that really scaled down sensitivity. And so it started already, I would say, modern way of seeing diagnostics. And in the 1970s, and actually I was surprised it was so early, it was the pregnancy test from my mom, I was studying in the 2000s. The 1970s, the first pregnancy test was developed. And I think FDA approved it at 77. And that was a big change of paradigm, because that brought for the first time diagnostics at home, diagnostics to be used by the patient. After that, I think we had like maybe 20 years that the revolution was the molecular test. So we are not anymore only seeing proteins and antigens. We can already detect DNA, RNA, pathogens, etc. And then COVID arrived. And what happened with COVID is that PCR and COVID antigen tests were mass produced worldwide and arrived in all the houses. And I think that COVID really, it was like the peak point for diagnostics. Everybody saw the need and how useful it is to have diagnostic tools, that they are fast and available to everyone. And nowadays, where we are is I think it's in the integration period. So basically we are producing complexity, bringing the test closer to the patients. Artificial intelligence is entering, is helping in decisions, is helping in image generation. There is the genomics. So it's we are opening an entire new dimension that will find the path through personalized medicine. So basically what I wanted to capture is in the last century, diagnostics has changed a lot, right? And it has moved from basic observation to molecular sensitivity to now personalized medicine. And how does the market, a diagnostics market, look like? I took this graph, I think it's from the report of last year report from Towards Healthcare. And basically what it shows is that at the global level, the diagnostic market is increasing. It's increasing, like in the 2021 was estimated to be around 75 billion US dollars, and is estimated to be like 125 billion US dollars in 2032. That's an increase of 70% of the market in 11 years. Not bad. So diagnostics is like growing and is growing to stay. And why is growing? I think it's growing because we have an increase of chronic diseases. We have like a population is aging worldwide, but also like because of the progress in the technologies like molecular diagnostics and point of care. But this graph also shows something else. And it shows that the market size is not the same and it's not growing at the same pace, depends on the region. Right, so let's look a bit more into it. And in this graph, it shows how it looks, the diagnostics market share per region in 2023. And what you can see is like most of the market is dominated by US and Europe. So two regions with very well-established healthcare systems. Southeast Asia and Pacific is growing, especially because large population, large demand, and also like an increase in investment. And Africa and Middle East is the one that is catching up, right? It's like emerging, is a field is emerging, and is expected to continue growing based on new investments and also a lot of technology transfers that are happening at the moment. And here there is a big opportunity, and I'm sure, and I really hope that in this room there is a lot of talent that will push the diagnostics in Africa for the future of diagnostics in Africa. And so we have seen how it looks in the market, who is leading, who is growing, who is catching up, but access to diagnostics not always follows the same trend than the market. And innovation, to me, is useless if it doesn't reach the people that requires those diagnostics. And this, it opens another thing, and it's probably the most concerning part, that is the access to diagnostics. So what is the diagnostics gap? Yeah. Consumers, like, yeah, is like the value of the market, the money that moves around diagnostics. Yeah, so there's this publication from Lancet in 2021 that they estimated in low and middle income countries for different diseases, what was the gap, the diagnostics gap. And the diagnostics gap, it means the portion of a population that has a disease that is not properly diagnosed. And they concluded that around 50% of the population that has a condition is not diagnosed. That's huge, right? And access, it's like we have the tools, and we will talk over and over this today. We have the tools, but we have to make sure they arrive to the people. And why they don't reach the people? Either the tools are not available, either they are not affordable, or they are not designed for the needs of the region that is going to use them. So how does these tools need to be useful? A diagnostics needs to be designed to fit the purpose, right? And that doesn't not only apply to low and middle income countries, that applies to all diagnostics. If you design a diagnostic tool that does not answer to the need, does not answer to the person who's going to use it, and where is going to use it, so the user and the settings, is going to stay in a drawer, right? And here I try to put like example. It might be a bit stupid example, but it's just imagine that you develop a test, let's say leishmaniasis test, and you're going to test it in Angola in rural settings. And your test needs cold chain and needs to be stored at 4 Celsius. But the place you go, there is no cold chain maintained. Then it's not going to be stable, it's not going to be useful. So always the test needs to be designed thinking where it's going to be. And that might sound very trivial. You might say, Olga, okay, that's obvious. But I tell you, most of the startups in diagnostics fail because, not because they don't have a good technology. They have cool technologies with very good performance. They fail because they are not designed to respond to a need. And there is a paper, it's very interesting, from 𝐂𝐀𝐑𝐄 Association, I think, consultancy, right? It was financed by Bill and Melinda Gates Foundation. And what they did is they wanted to understand why the diagnostics startups fail. And they had like 28 diagnostic companies, and they studied them for a while. And they studied what were their strengths and weaknesses along the life cycle of a project, since they were doing the R&D until they were entering to the market. And what they found that from those 28 companies, 15 at the day that they wrote the report, 15 they were already out of the market. And there were like 6 more that they call them zombies, that they were still around hanging there, but they were predicting they will not make it. So that's like 70% of failure rate. And why there was the failure rate? In most of the cases, it was because they were not answering to the need of the patient. So, so the in vitro diagnostics can be classified in 4 groups, depending on the technique that they use. So we have molecular tests, immunoassays, clinical chemistry, and cell-based tests. So the molecular test, they measure genetic material, DNA, RNA, and they can use different types of samples. They can dot in blood, oral swabs, muscle swabs, sputum, feces, etc. And this would be the PCR machines, for example, for respiratory panel that could put COVID-19, can also be tuberculosis test, and pox test. Many tests can be analyzed through PCR. Immunoassays, in general, they detect proteins or pathogens through the interaction of an antigen and an antibody. And it can also accept a lot of different sample types. And that would be like, is the technique used in the lateral? All right, so I think that clinical chemistry, we were talking about clinical chemistry. So most of the times it happens in blood, but it can also be using other sample types. And some examples will be when we measure cholesterol levels or iron levels. All the blood panel that we receive when we go to the doctor for the annual checkup, that's most of it is analyzed through clinical chemistry. And then the last one is the cell-based. That includes hematology and cell culture. And normally it's done in blood samples, but some other sample types can also be accepted. So this is based on the technique, but IBDs can also be classified through complexity, and that's what I find more interesting, and the path we are going to follow the rest of the conversation. And there is no one way of classifying IBDs through complexity. So in order to present it to you, I'm using the way that the categories we use at FINE, like the organization I was working before, and it created 4 categories. We are going to move from left to the right, and you will see that we are going to move from more complex to less complexity. And that also translates from less accessible to the individuals to more accessible. Yeah. So the first one are the laboratory IVDs. For these ones, it's like complex diagnostic systems. They require a lot of steps. They require external equipment like micropipettes, spinners. They need like technical people. Um, and, and in this category will be like the standard ELISA kits or ours. Then if we do one step and we make it less complex, we move to the category of that we call near point of care. The near point of care are desktop analyzers that require less steps. They need less training, but they still require some laboratory infrastructure around. Maybe you still need to use a pipette or a centrifuge. And here I put as an example the 𝐂𝐸𝐬𝐧𝐬 GeneXpert instrument. And we will talk about this later when we do some case studies. One step more, it will be the true point of care. These cases are fully portable, battery-operated systems that require almost no training and could be accessible to normally have disposable cartridges with all the reagents integrated, and can be, is probably the first type that can be decentralized from the laboratory and get closer to the people. And the last one, we call them instrument-free. That will be the lateral flow test. So it's like fully disposable devices that do not require power and that they are very simple to use. We are all very familiar to them, especially through COVID. So what I would like to do now is, if we go back to the healthcare levels, like in each level or in each setting, which is the type of device by complexity that can respond to the need. So if we think that we are gonna do the test at home or in the community, we have to think that we will probably should not rely on power. We should not need water or equipment, no temperature control, and the personnel is gonna be with no training. So in this case, if we go back to previous, if we go back to this slide, we will see that we need to be on the right hand, right? So like most simple devices are the ones that are gonna be relevant to the community. For primary care, still maybe the power is not reliable. So it's good if we still have systems that they are battery operated, but we can already increase a bit the complexity. We might have some laboratory instruments or supplies, a bit of like people more trained. So we can enter the near point of care also in these settings. And the final are the district hospitals, where there is like you can have all the complexity and the most advanced technologies, because the people that will be performing those experiments is going to be laboratory technicians well prepared. So basically the takeaway is that when you design an IVD, it has to follow the infrastructure that is gonna use, and not the way around. So yeah, and we just to put another example, I know I'm going to be very repetitive, but I think this is very key. If you want to use it, if you are designing a test to be performed at home and not have 20 steps with time steps, right? It has to be very simple and fast. Yeah, and from now on, as I said, laboratory diagnostics is a big piece of the cake, right? And it's very important. I'm not gonna contradict that, but I want to focus our attention in the point of care, especially because it's my field, but also because today I want to put emphasis in complexity and bringing the diagnostics closer to the patients. So from now on, we will leave laboratory diagnostics on the site, and we will focus more on point of care. And from the point of care, which do you think that is a system that is more used or more sell? So it is blood glucose measurements, blood glucose drips. And it has more than 50% of the market. And this is why, like, probably it is because of the rising numbers of diabetes worldwide, but also because it's a very well-established test for many years in the homes. And similar is for the pregnancy test, that it also, I think it's the next one that has the biggest market share. It's many years, it's very well established and works very well. When I saw this figure, I was a bit surprised of how little is the infectious disease point of care market. And I think it links a little bit with one of the first slides we discussed this morning. So infectious diseases are the main disease burden in the global south. But they are not the main disease burden in the global north. In the global north is the non-communicable diseases, like cancer, mental health, strokes. And if you remember, the market is dominated by the global north. And therefore, the point of care devices respond to their needs. Right? So what I expect is that with the increase of the penetration of the diagnostics into the global south, the need for infectious disease point of care is going to increase, and we are going to see this portion of the cake like up. Right. So basically, I think that we are in the best moment to be in diagnostics because we are really seeing an explosion of diagnostics nowadays. We are seeing many lateral flow tests penetrating like the homes and the communities, seeing molecular point of cares that will be deployed, change the way we test in primary care. But then there are also like fully automated, extremely sensitive, multiplex instruments in the laboratories. So I think it's very exciting to see how now the technology is available, how it's going to be deployed and integrated by societies, by healthcare systems, and by people. So I'm very excited. I think that you are in the best moment to enter this field. And before we wrap up, because it's so large, the diagnostics field. And of course, I'm not an expert in all of it, and we will be here forever. So what I wanted to do is give you like 2 different cases and talk a bit about it through 2 examples. So I will put examples of different products. And these are all examples. I have nothing to do with these companies. I'm not advertising these companies over any other product, right? So one case I want to study how using tuberculosis diagnosis, how it has changed the diagnostics with complexity. And then the other is I want to talk about home testing using lateral flow test. So the tuberculosis is a bacterial infection that affects primarily the lungs. And in accordance to the World Health Organization in 2023, it killed like 1.25 million persons. And there were 10.8 million of infections. So it is still one of the main killers worldwide. And the gold standard to detect tuberculosis is through the cell bacterial culture of sputum samples. And this can take weeks. Like, to have like a confirmed negative can take up to 6-8 weeks. And this delay on the diagnosis, it caused delay on treatment but it also gives a very large window for infections, for transmission. And it's very important to try to be faster in the diagnosis of this type of disease. PCR really did a big step. Now, using PCR, you have like results in few hours instead of weeks. But it still is limited to laboratories. It's still far from the patients. So the community is doing a lot of efforts in developing new technologies that they are faster, cheaper, closer to the patients. And this is what I want that we go through it with examples. So remember these slides. Basically, from PCR, we will be in the laboratory. And I want to show you an example of how we move to near point of care and another example of how we move to true point of care. So a PCR, probably a lot of you have done a PCR in the laboratory, so I'm not going to explain anything new, but you know it has several steps. It has some complexity. A lot of typing in the middle is time consuming. But the sample prep might take you half an hour. Then it depends of the assay and the instrument. And the real-time PCR might take you around 2 hours. So you still need to have, you need to be trained and you need to be in a laboratory. So that was like it's a big step from microscopy, but in culture, but this is still not a real point of care. And Cepheid, in 2019, it launched its division expert. And that product, I would say it revolutionized the tuberculosis diagnostic field. I don't know if you've ever seen it, but basically it's a laboratory benchtop instrument that works with a cartridge, a disposable cartridge that has all the reagents integrated in the cartridge. So the workflow gets simplified a lot. From those 7 steps we had before, we are moving to a very simple 4 steps. That is, collect the sample, load it in the cartridge, run the cartridge in the instrument, get the result. Right? So we have moved also from a few hours to 2 hours sample to result time. And the performance is quite variable. So we improve time without sacrificing performance. But the problem of the GeneXpert, well, the problem, it's not really a point of care system because it still requires a laboratory. It's quite expensive. I think an instrument can cost around 20K, not more, and each test around $15. But also like they have launched different cartridges for different diseases, so the platform can be leveraged to detect different diseases. But definitely it's not close to the people, right? But in order to go closer to the people, we need to go 1 step further in complexity. And this is an example of PlusLife. PlusLife is a molecular test that fits in a hand, in the palm of a hand. It is fully battery operated. It's isothermal. It uses isothermal amplification and fluorescence detection. It has a very simple workflow with a easy sample prep step, and the results from sample to the final report, let's call it like this, it's less than 30 minutes. So notice that we move from 6-8 weeks to 30 minutes. And we move from the laboratory to the primary care testing. So the evolution that is having like the technology and it's having like it's going to open a new way of diagnostic of diagnosis. Of diagnosis for this type of disease. Another thing I wanted to bring your attention is to cost. I said before, 𝐶 it's like around 14 maybe tests, and I heard it's not published anywhere, but I heard the TB test is going to be launched for $3.5 with the idea of scaling down, of lowering the price to $2 when they scale up production. So that's going to be interesting to see what is going to happen. Another interesting thing is if you want to get closer to people, you also have to think on which samples are you going to use. And this test, part of the sputum, is also validating tongue swab. And this is going to be very interesting for those people that they cannot expectorate, like elderly people or infants that they cannot give this type of samples. So that makes it like even more accessible to the real needs, right? And I'll use something. No, yeah, like this product, they have a COVID-19 test that is FDA approved, and recently, I think a few months ago, they got listed at WHO for the ERPD, so the emergency program for the TB, but they are still producing more data. And in terms of performance, we said the 𝐶 maintained the performance of the PCR. In this case, performance is expected to be a bit lower. We cannot have everything, no? Maintain performance, scale down, money, complexity. But it's quite close. But I'm sure that in the years coming, this is even gonna get better. And Plus Life is not the only company is doing this, right? There are many others like TrueNat, LumidaDX, several ones that they are in the process of getting approvals for this type of molecular true point of care testing. And it's going to be very interesting to see how the health systems are ready to accept them and how also the population is going to accept them. So we entered to the last case. And this is about lateral flow test. And I know these days you will be exposed to the amazing diagnostic scheme about lateral flow test. And I really encourage you to use this opportunity to be close to experts in the field, because lateral flow tests are probably the most used tests at home communities. And why is that? It's because they are fast, they are cheap, they are easy to scale up, they are simple to use, right? But of course, they also have their own limitations. I'm not saying lateral flow tests are perfect, because they have some limitations in sensitivity, they have multiplex limitations, and the results normally are qualitative or semi-quantitative. So there is always, and sometimes people think that they already arrive to the bottleneck for these limitations. But the way that technology is evolving is also bringing new solutions. And for example, nowadays we can see more and more solutions like portable readers, that they are small, battery operated, they cost less than $100, and that this will open, this opens a new opportunity to change the molecules that are used for the readout of lateral flow test. Normally you use gold, so you can see visually the result. But if we use a reader, then we can start using other fluorophores, for example, to increase sensitivity. So again, the technology is bringing new opportunities to improve performance of the techniques that we already have in hands. And now I'm thinking I could also put here like a picture of a cell phone. There is also like a lot of initiatives in using the cameras of cell phones to read lateral flow tests. In my opinion, this still needs a little bit more of work because it really depends of the camera, of the mobile phone, and the light conditions. But there is indeed a path in that direction. But one of the things that lateral flow tests really is self-testing. And self-testing, it gives opportunity to test at the commodity of your home, but that also means with privacy. And we know many self-tests. Wait, I've lost the track now. And yeah, and there is a global trend. There is a global trend through self-testing. So we see a lot of new products coming, like maybe to track hormones or vitamins, under the idea of self-monitoring of your own health. But there is another sector that really benefits from self-testing, and it's for all those diseases that they are in certain regions of the world, they might be submitted to a lot of stigma. So, for example, sexual transmitted diseases, maybe populations like sex workers or men who have sex with men, or people that inject drugs. They are in a very vulnerable place, and self-testing might give them the opportunity to be tested with privacy at home, and will increase the number, will reduce the diagnostic gap in these situations. And when you self-testing, I put 3 cases that everybody knows. One is the pregnancy test, the other is the COVID-19 antigen test, and then maybe it's not that well known, but it's important, an HIV test. And I would like that we think about commonalities between them, and also and pregnancy test, it predicts physiological condition, right? It's not a disease. Your life is going to change the positive, but you're not going to infect anyone. So it's safe. You can get your test at home. Everything is going to be good. For COVID-19, that's an infectious disease. But when you got a positive, what you had to do is to stay home. Take paracetamol, hope for the best. That's it. But what happens if you get an HIV test and you get a positive? You need medical follow-up, need medical treatment. So it's very important that the self-testing for infectious diseases, they come with a program where the person, if it gets a positive, is going to receive assistance. This is very important, and it's the main difference from the other ones. And this is also why in certain countries, there is a lot of debate about self-testing for this type of diseases, because they want to avoid panic, and they want to avoid self-medication. But again, if you remember what we just talked about, the stigma, in certain cases, in certain areas, in certain regions, the advantage of having these populations, having a safe space to be diagnosed, might overcome the risks of panic, of treatment. But it really needs to come hand by hand with a good program from the government to provide assistance. All of them are cheap because we said lateral flow test is one of their advantages, fast. And another interesting thing is the sample type. If they need to be used at home, it has to be samples that they are easily collected by us. The urine, nasal swab. We have, like for HIV, this is the oral quick. that induce the gums, HIV, or have a finger prick, right? But we will avoid samples like venous sample because we don't know how to extract venous blood ourselves, and we don't have the qualification, neither the tools. So when we go through home testing, it's also about which sample types we can. Yeah, and I think that this wraps up what I wanted to share today. I hope that it illustrated from what was the beginning to where it goes now, to diagnostics, what is the importance, especially when we work in diagnostics, to have in mind how this test is going to be used and which purpose, and how we can take advantage of all the innovative new technology to reach patients easily and affordable manner. Thank you very much. If you have any question or you would like to share your impressions, happy to answer. That's a good question. I hope so. That would be very interesting. Um, I think the lateral flow tests are gonna keep improving their performance. I think for a while we thought that it reached bottom, but now there are newer ways of detecting, newer molecules, newer techniques, newer materials, newer readers. I think that they are gonna keep improving sensitivity. But to me, the objective, it should not be that much to equal the laboratory performance, because there is more in place in the game, right? It's laboratory has its own role, but lateral flow test has its own going to the communities and to the houses. So it has to have the sensitivity that is relevant to diagnose the disease, but it doesn't, I think it's not a good approach to just be comparing them with the laboratory performance, because it really tries to answer to a different need. So I don't like the people that just analyze or compares technologies in terms of performance, because there is much more. There is like which sample type, how easy, how fast. So it's like so many parameters that we have discussed today that you need to take in account about how useful is a technique than just comparing performance. Not sure if that satisfies you. Thanks. Yes. You like That's a very good question. Okay. So I'm gonna repeat the question so I can check if I understood it well. So basically, you're asking what, for example, African diagnostic manufacturers can do to establish themselves in the market when there is little demand, little support from governments, and they have to compete with imported products. Was that your question? Okay. Well, maybe we can also ask Shah after, because he's a manufacturer expert. He has its own company here pushing the diagnostics field in Senegal. But to me, that there are different barriers, right? One is, as you say, demand. The local made products, they need to compete with imported products. And there is, even if the imported products sometimes might be expensive and not perfect for the settings, the population has a tendency to believe that imported products are of higher quality. And that's a change that needs to happen at the community level. And it's also in the hands of the manufacturer. They need to convince people. They put in the market high quality IVDs, right? Like the same standards. And also with regulatory approvals, they can show that they are the same standards, because they need to convince people to consume local made products, right? Another thing is price. One of the push for local manufacturing, I think, is at the end of the road, they need to be cheaper than imported products, because they will be made closer to the people, because you cut in distribution costs, intermediate people, license, labor might be different priced. But at the beginning, there are a lot of investments in making these facilities, in capturing talent, in technology transfers, that translates to a product that for a few years might not be that price competitive with an imported products coming from China, for example, right? So I think here is where, in my opinion, and I'm not talking for Africans, right? But governments and global procurers, they need to make a step forward. And they need to give a period of grace to local manufacturers where they commit to buy local made products if they are available, in favor to imported products, to make sure these companies can establish in the market, can gain the trust of the people, can be sustainable, and like groups like Global Fund is procuring most of the diagnostics for lower middle income countries. They should make this commitment. And the other thing is talent. If you want to grow the diagnostic field, you need talent. And this is what is in this group. And people that studying diagnostics, that is committed to stay in their countries, committed to advance the science, join diatropics maybe. and you know, and that also you have like the economical situation to attract those people that maybe left the boat and they got experience, they can bring it. I think there are many barriers to overcome, but hopefully it will answer. I don't know, share if we want to share. Yeah, start it.
If I haven't. So thank you so much for the presentation. I enjoyed every bit of it. Yeah, because am very interested in tuberculosis and diagnosis. So the question is that given that much of the funding for health innovations in Africa and Middle East comes from Bano, as you mentioned. So how can the local stakeholders advocate for more investment in diagnostic tools targeting antimicrobial resistance? Because you didn't mention this, and it's very important, especially in the case of gene experts. Yeah.
So you want to know about the funding and the trends on AMR? Yeah, yeah, that's a bit that's a good point. Good catch. I forgot to talk about AMR. Thanks for bringing this up. I mean, when you talk with scientists in the antimicrobial resistance, they see the future very dark. It's like, whoa. I mean, they say it's gonna be the new pandemic, right? Like more and more resistance are rising, and there is a big concern about misuse of antibiotics, but also the impact we are having as humans to the environment and to the animals is also translating to more and more resistance. So I think that now is bringing a lot of attention. There are a lot of organizations worldwide that they are exclusively financing diagnostics for AMR. And for example, in STIs, I've seen many products that they do gonorrhea and test for resistance to gonorrhea, for TB. Like one of the critics from the true point of care Plus Life is that it doesn't do resistance. CFI does resistance. So it is something that definitely will be coming in the next years because it's a global concern for everyone.
So, the translator, considering the essential drive toward decentralization of HIV diagnostics, moving from central to community point of care settings, and the need to maintain patient privacy and data security in environment with social stigma, like almost every African country, especially in West Africa. How can African scientists be best influence and contribute to the manufacturing of, especially in infectious diseases like HIV? How can they contribute to the manufacturing of diagnostics? Yeah.
I summarize the question about the manufacturing. How can we sustain? How can we?
Yes. All right. So if I understood well, what you want to say, like, okay, like the community is already in this stigma situation, and as a scientist, you wanna do your research, for example, in sexual transmitted infections, and how do you raise the trust from the people to be part of your studies and use it, right? Um, I- maybe this answer is not gonna be very good. I'm just like trying to formulate it well. I don't think that is the responsibility of the scientist, right? I think this is the responsibility of your group. You need campaigns because it means a change of society behavior. It means just a change on mentality. And sometimes it's very difficult to change the mentality of older people. I'm just like entering in a very dark place, but older people. But I think that you're like younger societies that you really see these situations. You really see that there is people in a very tricky situation, and you see the need. You are already doing these steps of changing the behavior, but you really need the support of the governments. You need, or maybe from the institutes, make campaigns to explain like the advantages of this type of testing. Use the news. I think that public convincing is like is very complex. Change of entire society. Like, so I think that again, all these projects in diagnostics, when I was studying, they were very feel like they were very specific of a field. You are a microbiologist or a microtechnology, and you are doing your diagnostics. But this is not the case anymore. I think that now it needs a very broad amount of people and experts, because you need the technical people, the scientists that will push the technology, but you also need regulators there. You need like the anthropologists helping you. You need the clinicians helping you with the campaigns. So I think that you need a lot every time each to advance the technology, you need a group of experts to make it happen and the access. And especially in rural places, right, it's gonna be more difficult. But I will be here for the rest of the day, so I'm happy to continue the conversations with you and spend this time with you. Thanks for coming. Thanks for your attention.